thank the organizers of this meeting, the sponsors, the panelists. This is a real, testing times for us, owing to the COVID-19 situation. But I think this platform, this virtual platform, is still kind of a good venue for us to kind of get together and discuss some of the most exciting, programs and projects in the biotech area. And so thank you once again for having me speak in this meeting. So I'm going to be talking about BERG's AI approach for target mapping and understanding disease biology and target biology. And then I'm going to give you one example of a novel target that we took from concept all the way into right now in IND enabling studies and into IND next year with the goal of entering first in humans in 2020. So this is our forward-leaning statement. So I'm going to start with the BERG's AI platform or AI approach. I know AI is an overhyped term, and there are a lot of flavors of AI these days. And just to be clear, we are not AI in the computational drug discovery space. We are more AI in the target biology and human phenotypic, we call it internally. And so just to start off and just kind of lay the table for discussion, what you see here is two sets of schematic. One on the left that talks about the traditional approach of how do we go about discovering targets, drugs, and then take it through the drug development process. A lot of it is based on literature or information in the public domain on genetic genomic data that's out there from various lines of evidence, including preclinical evidence. What BERG does is unique in many ways in that we put the human biology up front. What you see there is a silhouette of a human with the digitized version, or you can see the binary numbers, suggesting that we take the human biology very seriously and we put it up front in our discovery process. And so pretty much anything and everything we do from that point on is governed by our discovery in human cells, human tissues, human plasma, human fluids, and so on and so forth. And so we identify targets based on decoding human biology using AI, or I'll refer to as Bayesian mathematics or Bayesian algorithms to understand causality. And from there, we take the target into a discovery development paradigm, and in fact, validate the target or revalidate the target back in humans in a phase 1 setting. So that is kind of the distinction between what BERG does, putting the human biology up front and the patient biology up front in identifying targets that are clinically meaningful and are triaged at various points in the development based on the various risks of developing a particular target and a molecule. And so just to walk through, we call the AI process as the interrogative biology process. We interrogate the biological system. And as you can see here, this is a panel of various activities that lead to the identification of various targets. On the left is the discovery models themselves. And so the emphasis for BERG has been to collaborate with some of the academic hospital centers over the past many years. We've been around for about 10 years now in collecting and harnessing some of the well-annotated human samples that are out there for various disease indications, whether it be oncology or neurology. Here is just one example of how we use some cellular models, and then what we subject these samples, as you can see on the second, the biological capture or multiomics that includes genomics, proteomics, lipidomics, and metabolomics. Again, this is not like a cookie cutter thing. It's very specific to the project itself. There are certain projects that demands the use of proteomics in various flavors, phosphoproteomics, ubiquitination omics, and various other post-translational modifications. Similarly, lipidomics comes in structural lipids and functional lipids, and metabolome has its own diversification as well. But primarily, we try to capture as much data as possible based on the questions that we are asking in that second layer of harnessing or gathering information, or biological capture, as we call it. The third piece is very critical. It's not called what we call as knowledge engineering, where we actually use the data to learn from the data itself. So there are no prior inputs or prior knowledge that goes into the making of these models. These models are based on Bayesian causal inferential methodologies, where one event leads probability of one event leading to another, and so these ensemble of models are basically created using this iterative process of finding out what are those connections between various molecules and clinical readouts. So it can be, for example, progression-free survival or resistance refractory to a particular drug. So what are the key drivers of that particular phenotype? And so phenotype becomes absolutely critical for us. And so that, as I said, is what we call that the AI algorithm as basis. Primarily, it does not take into account any prior knowledge or biased information. It basically creates these networks from data itself. And I want to be very clear about that. And then, so from there, we create networks for both, causal networks, as we call them. Causality is the underpinning of what we do. Causal networks lead to slices of various data networks, as you can see. Number 4, we are able to create these slices of networks from this comparison, and then from the networks we're able to create rank order lists of targets and biomarkers, and then targets go into target validation, biomarkers go into some sort of like a biomarker validation. So that is the underpinning of what we do at BERG, the AI platform, and what we call as interrogative biology. And so from this process, we have identified a handful of targets, as one can imagine. There are 88 targets in oncology, 51 in neurology, metabolic disease, vascular biology. We have focused primarily on oncology and neurology in the last many years. I would say 4 to 5 years. Our batting average, a success rate for target validation is 77%. And what I mean by that is the recapitulation of the predictions of the platform, connecting omics proteins to an outcome, is what we calculate as a success rate. We are able to take those connections into a wet lab and revalidate them through a knockdown or overexpression experiment. And so out of the 31 targets that we have looked at so far, 24 have, and that kind of goes to the 77%. Out of those targets, we have taken 6 of them forward, 4 of them in collaboration with the various government agencies, including ORNL. 2 of them actually are in-house and are, as I said, are moving towards IND and first in human sometime next year, 2021. So as a next step in this process of benchmarking the output of the platform, we leveraged the study that came out in 2019, probably one of the largest functional genomics studies that were run in the past many years. The Behan et al. Paper that came out in 2019, looking at CRISPR-Cas9, synthetic lethality screening of various cancer cell lines, and so based on that, we were then able to take the what we call as Behan targets and basically overlay the BERG cancer targets, or the BERG cancer model, and we got quite a bit of unique targets out of the process. About I would say 20% of the targets were unique, and 80% kind of overlapped with the Behan et al. Study, the CRISPR targets. And because these are functional genomic studies and they come with functional information on the extent of the effect of the target on proliferation and various phenotypes, we were able to then rank order and prioritize these targets internally for our own discovery development efforts. So as I said, there are 46 overlapping targets, and out of which we could take few that were very unique, and we thought would merit the development of candidate molecule and drug molecules for developmental activities. And the 20-some%, like what I talked about, is very unique and not overlapping with the Behan et al. Study. So that we have kept aside for further target validation. So that's one of our recent case studies of how we benchmark the outcome of the BERG interrogative biology process. The next few slides, I'm going to talk quickly about one such case study or an example of how we took a target from target ID to drug discovery, now candidate selection, and now it is actually in IND enabling studies. This is a target from the ubiquitin proteasomal pathway. It's an E2 enzyme, the ubiquitin proteasomal pathway. And again, going back to the BERG platform, basically all the four to five components I talked about earlier are here on the slide as well. We started with cells for this early project. This was done very early and back in the days, where we started with cancer cells and used various conditions and perturbations along with certain environmental mimics of high glucose, low glucose, lactic acid, normoxia, hypoxia, and collected omics data, proteomics data primarily, uh, along with some functional readouts, like the metabolic fluxes and proliferation and growth and so on and so forth, which is in the biological capture. Did the knowledge engineering using basis, creating the causal connectivity map, as I said before, creating the data slices in number 4, and from there creating the ranked order list of targets based on the connectivity on that map. And this particular target, BPM 42522, I'm not revealing the name today, is one of the ubiquitin proteasomal targets of the E2 enzymes that I was talking about that we took into target validation. Now, what the network methodology allows us to do is also glean information on what are the closest connections in terms of protein function and gene function. And we paid close attention to that. So primarily for this particular target, BPM 42522, the nearest neighborhood of other proteins that are interacting or causally connected, I should say causally connected, not interacting, to this particular E2 enzyme are involved in the regulation of microtubule cytoskeleton, T-cell antigen receptor signaling, nucleosome histone function, cell cycle regulation, and apoptosis. So these are some of the inferential functions or phenotypes that we could glean out of this network approach for BPM 42522. So we moved on with the target validation of this particular target. Here's a panel of some data. On the left, on the top, we did siRNA of this particular target in Miyapaka, and a bunch of other cells, SK-HEP1 and HEPG2, where we actually saw a good effect on cell proliferation. Whereas some cells didn't respond to the siRNA knockdown, very clearly showing that there is some sort of selectivity for the target itself. The right panel on the top, where we carried out some siRNA experiments, we confirmed that the effect on proliferation comes from an effect on apoptosis, and there is a pro-apoptotic effect that you trigger with the knockdown of BPM 42522. The bottom two panels on the left, we actually confirmed that the proliferative effect or the effect on viability is also coming from an anti-proliferative effect or a cell cycle effect, primarily a G2 to M cell cycle arrest that we are seeing in these cells. And then just for a quick ex vivo validation, we looked at the expression levels as well as localization of BPM 422 in pancreatic cancer patient tissue slices compared to adjacent normal tissues. And we actually saw there was an increase in the expression of this target and actually an increase in localization to the membrane in pancreatic tissues, cancer tissues compared to adjacent normal tissues. So that was the extent of target validation we performed. We did additional studies on understanding the MOA of the target itself. But the next few slides, I'm going to quickly move on to the drug discovery efforts that we undertook. Because this was a novel target, we used a fragment-based drug discovery approach towards identifying hits and going from hit to lead. Primarily what you see on the schematic on the left is the various molecules that were advanced. We identified a novel pocket in the whole process, a novel allosteric pocket that I'm showing on the right. And then there are some precedents for this kind of allosteric mechanisms in E2s. What you see there is this E2 homolog called CDC34 that was published several years ago, and the identification of a novel allosteric modulator CC0651 that is published in Cell. So there is some precedence for allosteric regulation in E2s, and that we thought was probably the best way to pharmacologically modulate this target. So we developed a whole series of biochemical, biophysical assays, actually showing there that what you see is the molecules having an effect on discharge of ubiquitin on the left top. We did some selectivity panels for UB2K along with other E2s, showing that this is unique to this particular target, BPM42522. We also did some biophysical SPR type of assays to confirm binding of these moieties, these molecules to the target BPM42522. So that was the extent of biophysical, biochemical characterization / selectivity, specificity assays that we ran for these lead molecules. And finally, we arrived at a point where we could test these molecules in cell potency assays. And what you see here on the left is some molecules were very potent, others were not. And we now understand why that is based on a mechanism, a differential that we know causes the anti-cancer effect. And again, these molecules are able to recapitulate some of the effects of the target knockdown itself, which I think is very, very confirmatory that this is mostly on-target activity that we're seeing. We do see the G2 to M cell cycle arrest for the cells that were treated with these compounds or these lead molecules. These lead molecules have no effect on other proliferating cells, which is also an encouraging sign. What you see there is the work experiments we ran with PBMCs showing no effect on proliferation. And then these were orally available molecules. We did quite a bit of development work on the oral bioavailability of these molecules for a xenograph model. And what you see on the right bottom panel is in two xenograph models, we were able to see quite robust tumor growth inhibition of over 60 to 87% in a dose-dependent manner for some of these advanced lead or optimized lead, as they are called. So right now, the plan is to advance these orally available molecules into IND-enabling studies and eventually into IND. The key here is that the phenotypic recapitulation that I was talking about and on-target engagement, we actually see that these molecules have an effect on cell cycle, as I had explained previously. There's an effect on G2 to M for sure. We see a robust change in cyclin expression, as well as some of the markers of cell cycle, namely phosphohistone H3. So we clearly see an effect on cell cycle and apoptosis inferred from the causal connections based on the network topology that we ran. And so just the message together here, the point is we have a clear understanding of mechanism, we have an understanding of which of the tumors and what kind of clinical setting would these molecules have the most benefit or the most effect. And so that kind of paves the path for translation for us. And we have some entry points into phase 1 as we're looking at phase 1 using the platform itself. And so as next steps, I mean, as you can see here, these molecules are potent across a panel of cancer cells. Various cancer cells are showing potency for these molecules, allosteric modulators for the target BPM42522. And we have developed them to a point where we now have good DMPK profiles, good tolerability. We have a formulation now developed for both rats and dogs that we're taking into toxicology studies and IND-enabling studies. And so just to conclude, I guess I have 5 minutes, maybe 5 minutes for questions. What I hope I could showcase today was an example of using a network biology approach and AI to map targets and to take one target from concept, from target validation all the way into drug discovery development and phenotypic recapitulation based on the inferences from the platform or the network topology approach we took. We are actually in the process of leveraging several more data sets around various cancers, tumors that we have access to through collaborations on pancreatic cancer, breast cancer, and so on and so forth, to kind of help us guide the clinical development process, identify which patients would benefit from these novel therapeutics and novel molecules. And what I didn't talk about today is there is also along with the targets, the networks also encumber several markers and biomarkers of patient stratification. So that is in development as well. That will kind of support the label claims and TPP. And then we also have some follow-up best-in-class molecules in development. There are other targets that are also in early hit to lead stage that are also equally exciting. But I think that this is the full extent of what we do as an enterprise, as a company, using AI in the front end to identify targets and in the back end to identify patient populations, and then take more of a streamlined approach towards drug discovery and development. With that, I'll take questions. Thank you for your attention.
Wonderful. Thank you so much, Vivek, for your presentation. And we do have a question for you from Andrew, Vice President of Rappel's Pharmatech. And he's saying, hi, Vivek. The knowledge engineering sounds very interesting, as you mentioned. The system sounds self-learning. I'm curious if there has been any testing of the system on some historical data and seeing if it predicted known real-world results.
Absolutely. So we work with several large companies. This knowledge engineering platform has been used in context of several large companies, and there are pre-existing data sets. Details, but there was a flu vaccine project that we got onto using knowledge engineering. There are several other examples with government, non-government, where we have used the knowledge engineering piece to kind of self-validate and learn in the process. And we have had some astonishing results.
Wonderful. And there's another question for you from Larry Hardy. He said, Vivek, nice description of your progress in oncology. Are you similarly advancing in neurology?
Absolutely, good question. Interestingly, this E2 target has a role in neurology as well. And we have reached a point where there is a divergence in the pharmacology that allows us to go down that path as well. So there is a follow-up neurology program that is coming right behind this program. Well, Larry.
Wonderful, wonderful. And yeah, as you all know, Vivek, he will be in the future also speaking at our in-person gatherings in Boston and Cambridge. In the past, my team and I have worked with him, and he's a very, as you guys know, very highly anticipated speaker, partially due to a lot of his platforms that he has developed. So once again, Vivek, thank you for your presentation. And if anybody's interested in meeting with Vivek, do let us know.
Thank you all.